السلام عليكم أعزائي الطلاب. This is the second lecture regarding amino acid metabolism. We knew from the first lecture that we have an amino acid pool around 100 gram, and this pool came from either ingestion of proteins, degradation of internal proteins, or synthesis of non-essential amino acid de novo. This amino acid pool is used for various purposes, sometimes for synthesis of structural protein or for different proteins, sometimes for synthesis of specialized product such as purines, pyrimidines, creatine and others, and sometimes we use this amino acid pool to gain energy, and here we need the catabolic pathway. The first step in the catabolic pathway is to deprive the amino acid from their protection. What protects the amino acid from the catabolic pathway is the alpha amino group. Each amino acid contains at least one amino group, which is the alpha amino group. The first step in removing this alpha amino group is transamination. It is a reversible reaction involving the action of amino acids with a keto acid, most likely alpha ketoglutarate, to form glutamate mainly and the corresponding keto acid. So the main purpose of transamination is to funnel the amino groups from different amino acids into glutamate. In this step, there is no net loss of amino group. We still have an amino group. Rather than different amino group, we increase the amount of glutamate. So now we need to remove this amino group mainly from glutamate, and here comes the rule of deamination reaction. Deamination reaction is of two types. The first one is oxidative deamination. From its name we know that it requires the presence of oxygen, so it's only function in aerobic conditions. And the non-oxidative deamination not requires the presence of oxygen. The oxidative deamination is the most important one, and we all should know it. Non-oxidative deamination, it is better not to know, but it is not very important. Okay, after this we will discuss ammonia metabolism in a brief. The deamination reaction is of two types, either oxidative or non-oxidative. Both of them involve the directly removing the amino group from the amino. Deals mainly with the glutamate, so it is the most important of this reaction. Involve the oxidation of glutamate to its corresponding skeleton, which is alpha ketoglutarate, catalyzing by an enzyme called glutamate dehydrogenase. Glutamate dehydrogenase, from its name we know that it involves removing of hydrogen or proton, so it needs a coenzyme with a hydrogen acceptor. This is NAD. So glutamate dehydrogenase with its coenzyme NAD will remove the amino group from glutamate, forming alpha ketoglutarate and NADH. So, in general, different amino acids by transaminations would be converted into glutamate. This is the forward reaction. And this glutamate by oxidative deamination is converted into alpha ketoglutarate. This is accompanied by production of ammonia and require conversion of NAD into NADH. So this is the forward reaction in which different amino acids will be converted into alpha-ketoglutarate by the combination of transamination and oxidative deamination, and with the result in the production of ammonia. Is involve the combination of ammonia with alpha-ketoglutarate by the same action, by the same enzyme, by glutamate dehydrogenase, but requires the conversion of NADPH into NAD, and this results in the formation of glutamate. Glutamate by transamination will be converted into different amino acids. Of course, these amino acids are not essential. We cannot synthesize essential amino acids. So alpha-ketoglutarate, this is the anabolic pathway. This is the pathway that occurs when we require the synthesis of different amino acids. So this is the anabolic pathway, while the previous was the catabolic pathway. So both transamination and oxidative deamination occur as a link, as a common pathway for anabolism of amino acid and for catabolism of amino. This is the oxidative deamination. We saw that the oxidative deamination mainly takes place with the glutamate. Is there any other oxidative deamination? Yes, there is oxidative deamination, not explained in these slides, involve what is called D-amino acid oxidase. Most of amino acid in our body is L-amino acid, while amino acid presence in the plant are of D origin, of a D isomerism. They are D amino acids. When we ingest plants, we will have proteins, and these proteins catabolism results in the production of D amino acids. In the intestine, we have an enzyme called D-amino acid oxidase. D-amino acid oxidase directly oxidize this amino acid into corresponding keto acid with the production of ammonia. This occurs in the intestine, and this is the other type of oxidative deamination. The non-oxidative deamination involves the conversion of other amino acid, for example serine, threonine, which we saw it is not involved in the transamination, cysteine. Both of them are corresponding by specific enzymes called lyases that require pyridoxal phosphate. أيضا مثل الترانسامينيشن عندنا non-oxidative deamination يحتاج pyridoxal phosphate. So B6 is very important in oxidative deamination. The enzyme serine dehydratase converts serine directly into pyruvate without the need of transamination, with the direct production of ammonia, and it's called dehydratase, not dehydrogenase, so it not requires the presence of oxygen. Now, in both oxidative and non-oxidative deamination, there was liberation of ammonia. Ammonia is very important, and it is a very harmful substance. Why? Ammonia can be converted into ammonium ion, that means NH3 converted into NH4 plus. NH4 plus have a charge, so it may affect the acid-base balance. Also, ammonia is toxic to the brain. Ammonia is toxic to the bone marrow. It inhibits the synthesis of RBCs. So it is very important to produce ammonia in a regulated way, and the only safe way and the only safe place to produce ammonia is in the liver. So ammonia may rise from different processes of ammonia. From action of amino acid oxidase, we speak about in the intestine. Amino acid oxidases will result in the direct production of ammonia. From non-oxidative deamination, from hydrolysis of alpha-ketoglutaramic acid and alpha-ketosuccinic acid, this is not very important, from hydrolysis of glutamine by glutaminase. We know glutamine and asparagine, both of them containing two amino groups. So by asparaginase and glutaminase, glutamine is converted into glutamate with direct production of ammonia. This is also an important source of ammonia. Sites of the body and most tissues. And so, how can we dispose of this potentially harmful ammonia? The first way is for interaction with an alpha-ketoglutarate to form glutamate by glutamate dehydrogenase. We know that glutamate dehydrogenase can convert alpha-ketoglutarate into glutamate by combination with ammonia. So, of course, this is not the ammonia produced by the same enzyme. It is non-logical to say that we have glutamate dehydrogenase, we have oxidative deamination, which produce ammonia, and then the same enzyme combine it with alpha-ketoglutarate to form glutamate again. This is not logical. So this ammonia that's produced from other sources such as amino acid oxidase or from non-oxidative deamination can be combined with alpha-ketoglutarate to form glutamate. Glutamine can be synthesized from glutamate again. Glutamate combined with NH3 by the enzyme glutamine synthetase, which requires energy, requires ATP to form glutamate. See, this is in most tissues and mainly in the brain. Ammonia, which is produced, can be combined with the glutamate to form glutamine by the enzyme glutamine synthetase. Glutamine is non-charged. It can be transported in the plasma when it reaches the liver. It undergoes two steps. The first step is by glutaminase. We remove the first amino group, converting glutamine into glutamate. Then by oxidative deamination, by glutamate dehydrogenase, it is converted into alpha-ketoglutarate, and we have produced 2 moles of ammonia. So this glutamine helps to transfer 2 groups or 2 moles of ammonia from most tissues into the liver, and the liver is the only organ that can handle ammonia effectively without any harm. It's mainly converted into urea, which we discuss in the next. This is very important. We say that the first route to deal with ammonia is converting it into glutamate by combination with alpha-ketoglutarate. The second one is converting it into glutamine by combination with the glutamate. And the third important way of disposal of ammonia, and the most important way, is by synthesis of carbamyl phosphate. Carbamyl phosphate is an important product or intermediate product that is involved in the synthesis of urea, which we will discuss in the next lecture. CO2 plus ammonia with 2 ATP by the important enzyme carbamyl phosphate synthetase carbamyl phosphate will be formed plus 2 ATP and Pi. This enzyme carbamyl phosphate synthetase 1. We have 2 types. We have carbamyl phosphate synthetase 1 and carbamyl phosphate synthetase 2. I want you, student, to look for these 2 important enzymes. What are the differences? Well, there are 3 important differences between carbamyl phosphate synthetase 1 and carbamyl phosphate synthetase 2. I may ask you about the comparison between these enzymes. This is a 3-point comparison, and it is very important. You should know it. Okay, this carbamyl phosphate mainly formed in the liver, because in liver there will be production of urea, and this urea is the ultimate way to deal. Okay, last subject for this lecture is to speak about what is called the glucose-alanine cycle. What is the glucose-alanine cycle? its cycle occurs by liberation of glucose or transport of glucose from liver into muscle and different tissues, and transport of alanine from different tissues into the liver by these steps. First, protein degradation in the muscle when there's an exercise or different muscle any movement of muscle. In fact, there's even in the static state there will be degradation of proteins of muscle. So protein degradation in muscles results in the release of different amino acids. In general, these amino acids will have the amino group of this amino acid will be funneled into glutamate by transamination. Here it is not safe to transport glutamate from muscle into the liver. Why? Because the glutamate is an acidic amino acid. So rather than transport glutamate directly, glutamate by ALT is converted into alanine. The amino group from glutamate is combined with pyruvate, which is also produced in aerobic conditions in the muscle, only in aerobic. In non-aerobic conditions there will be no pyruvate. We know there is lactate, so this glucose alanine cycle only functions in aerobic conditions. The amino group of glutamate is combined with pyruvate, result from aerobic respiration by ALT. This glutamate, pyruvate, and yes. So the end result of this first part of the cycle is that protein degradation results in the increased concentration of alanine inside. This is muscle proteins degradation forming ammonia. Ammonia is funneled into glutamate, and then glutamate by transamination with pyruvate forming. Now alanine is transported in the bloodstream to the liver, and in the liver it is converted back into pyruvate by ALT by the reverse of the previous reaction. Here alpha ketoglutarate combined with alanine also by ALT to form pyruvate and glutamate. Why? Here glutamate of course will be by non-oxidative deamination converted into alpha ketoglutarate, and the ammonia is involved in urea synthesis in the liver. The pyruvate, which is produced by this reaction, is converted into glucose by gluconeogenesis in the liver, and this glucose is secreted into the bloodstream to reach the muscle to continue the cycle. So, presentation: muscle proteins forming ammonia, and then glutamate is converted into alanine. Alanine transported in the bloodstream into the liver. In the liver, alanine is converted back into pyruvate and glutamate. Glutamate involved by oxidative deamination and the synthesis of urea to get rid of the amino group, and pyruvate by gluconeogenesis is converted into glucose, which is backly secreted into the muscle, forming this cycle of glucose, pyruvate, alanine, and this is called the glucose-alanine cycle. Only function in aerobic conditions, because only in aerobic conditions there is production of pyruvate rather than lactate in the muscle. This is other representation. This is the same cycle, but this is better. We see it here as a cycle. Glucose in the muscle converted into pyruvate, converted back into alanine, alanine transported from the muscle to the liver, and the liver alanine lose its ammonia group to urea, here of course by oxidative deamination, converted first into glutamate, and the glutamate will lose its amino group, and alanine is converted into pyruvate. Pyruvate by gluconeogenesis forms glucose. Glucose secreted from the liver to the muscle to produce a continuous energy for the muscle. So the glucose-alanine cycle helps to produce glucose, to give glucose for the muscle for continual exercise, and it also gets rid of the amino group and the alanine and the degraded products of the muscle proteins.